I'm Cole Mathis. I'm a PhD student at Arizona State University. I'm a theoretical physicist. I study the origin of life, and I just got back from 10 days of field work with geochemists in Yellowstone. I already mentioned the title of my talk. Before I get started, I want to thank a few people. Everyone for Me Life is awesome. They're a research lab. They're great. These two schmucks, constant sounding board for me. They're both here. I appreciate their input on all these crazy and half-formed thoughts that come out of my head. Some people that aren't here, Wim Hordek, Lido Peel for introducing me to generative models, and Niles Lehman, who does autocatalytic sets in RNA chemistry, has had some interesting input. My advisor, Sarah Walker, she's spectacular. She's super awesome. Oh, and the Snake River for nucleating the idea behind this talk in my head somehow. Also, if you're wondering what a theoretical physicist does for fieldwork, that's about it. It's very theoretical. All right, here's a slightly better name for my talk. So I'm going to talk about the emergence of dynamic community structure in maybe autocatalytic networks. So hopefully I can unpack what that means by the end of this talk. It's a complicated idea, but I think with excellent introductions from Harrison and Ben, it'll be easier for everybody to swallow, and hopefully I'll understand what I'm saying. All right, so if you study the origin of life, eventually you ask yourself this question, right? And Tessa touched on this earlier. What is life? We don't have a good answer. But there's some features, right? So one feature of life that you might want to try to explain is all of the diversity we find in the biosphere, right? That's one thing that's like, wow, how do we explain this diversity that's everywhere? How can we come up with a mechanism to really handle that? Another thing you might be curious about is like life is actually this very ordered and structured sort of set of chemical reactions that have a particular function and operate in a particular way. So these are two different interpretations to this question. They're both obviously valid answers, but depending on what you think is more interesting and more important, you might go different ways, right? So if you're interested in explaining diversity, you know, our friend Darwin told us there's a way to explain this diversity. It involves things that make copies of themselves and a few other features. You might study things that do this. In this talk, I'm going to focus on the kinds of things Ben was talking about, which are autocatalytic sets, which are coupled reactions that form networks, and sometimes these networks have autocatalytic features where they can make more of themselves and everything in it grows exponentially. So that's what I'm going to focus on today. So here's a brief outline. I'm going to describe my model, which is very similar to the one Ben described. And then I'm going to talk about some background in autocatalytic sets, some results from the early 70s, and some later refinements. I'm going to mention networks, and in particular, I'm going to talk about stochastic block models as a type of generative model. And then I'm going to talk about a little bit of information theory, describe mutual information. It's easy, don't freak out. And then I'm going to show how to identify structure in the dynamics of these autocatalytic sets, and maybe some hints that there's a phase transition to dynamic order in these things, which is rampantly speculative, but I think I can justify. All right, so my model is really similar to Ben's. So there's this sort of background chemistry going on, right? There's gray squares and blue squares, and they can come together. They can form these sequences. Gray and blue could be 1 and 0. They could be A and B, whatever you like. So there's ligation when they come together. There's dissociation when they fall apart. In my model, in contrast to Ben's, it's closed mass. And I explicitly model the degradation. So that has some interesting feedbacks, but it's very similar. So all of these reactions for all possible combinations of sequences are possible in mine. And then there are some catalyzed reactions. So this is an example of the formation of this gray dimer could be catalyzed by the gray trimer, right? And there's lots of different ones you could do. I'm going to spend the entire talk talking about this particular network of reactions. It's sort of obvious when you look at it. It's a mirror image. Make dimers, make trimers. Trimers make these. Trimers help things that go astray. Right? So pretty simple. Is anybody confused by this yet? Cool. All right. So background on autocatalytic set theory. So if you take all of these reactions and you randomly sprinkle catalysts, you say this molecule catalyzes this reaction, you're guaranteed to get autocatalytic sets. If they're random, you're guaranteed to get autocatalytic sets as long as the probability is high enough and as long as the polymers are long enough. This is a result from 1970. A lot of chemists got really mad about it. There's been a lot of refinements. It's held up for 40 years at this point. Here's a great reference if you're curious about these kind of things. Wim Hordek studies them extensively, and he's really good at handling them. So now let's take a step back. So keep this in your head. Keep this in your head. here's another thing to keep in your head. All right, what are networks? Harrison did a good job describing these things. Here's some example of networks, right? Nodes and edges, an entity and a link between entities, right? How do we describe them? One way to describe networks is with what's called an adjacency matrix, right? So like this network, you have 1, 2, 3, and 4 here, right? We could label 1, 2, 3, 4, 1, 2, 3, 4. We could put a 0 if there's no link, and a 1 if there is a link, right? Simple description. And then you could get different combinations of these to form different matrices, right? So these are two representations of the same thing. Similarly, we could weight the edges, right? So this is a slightly different kind of network, but all we've done is instead of 1s and 0s in this adjacency matrix, we've put real value numbers, right? It's a slight generalization. Okay, so now that's in your head. How do we describe networks? One way to describe networks is with a thing called a generative model. There's lots of different kinds of generative models, and the idea here is you have a statistical representation of the network. You define a set of parameters that you can use to generate networks which are statistically similar to the one you're interested in. So for example, here, we've got 3 different networks. They all have 4 nodes, and they all have, what, 4 edges? Yeah, maybe more. I can't count. So the idea here is these are all equivalent. They all have the same number of nodes and the same number of edges. So I could make a generative model that says, I'm going to describe my network by the number of nodes and the number of edges. And you could refine that a lot further, right? So generative models describe structure in a statistical sense. That's the key point. All right. I stole these slides from a professor here at CU Boulder, Aaron Clausette. He's pretty rad. He put them online for free. I don't think he'll mind. They're great. Right. A particular class of generative models for networks that have become very, very popular are called stochastic block models. Stochastic block models look for community structure in networks. So computer scientists mostly worry about these kinds of things, and computer scientists like well-formed problems, right? So here's a well-formed problem. I'm going to give you guys a social network for everyone in this room, right? And then you're going to tell me if there's community structure in it and what that is, right? And so we know there's community structure in here, right? There's like a bunch of us from ASU. There's all these CU people, right? So this might be our social network. Lots of us know each other. But these might be the communities within it, right? This might be ASU. This might be Boulder. This might be somewhere else. This might be everybody else, right? So you want to break the network into structures where within the community, their properties are similar, and without the community, their properties are different, right? It's a simple idea. There's a lot of different ways to do it. This is a associative network, right? So where things that are similar associate with each other starts to look like this. This is the inverse of that, dissociative. You don't hang out with anybody that's like you, right? This is what you should do at a conference, right? Don't hang out with the people from your lab. Go meet other people. There's ordered ones where it's kind of in between, and then there's these core periphery ones, which are of particular interest to metabolisms. All right, so what have I told you? I've told you about my model. I've told you about a generative model for networks, right? We can describe the structure of networks in a statistical sense by creating a model that makes things like it, right? All right, everybody got all this in their head? I'm going to add another piece. Here's information theory. This is like page 2 of an information theory textbook. It's not scary. It's called mutual information. If you have 2 variables, x and y, the mutual information between x and y is how much do I learn about y given that I know x? Or how much do I learn about x given that I know y? It's symmetric. It's totally correlation. There's no causal anything. You could think of it like a linear correlation except super generalized, right? So it's kind of like an r² value for things that are like so far away from linear you couldn't even fathom. All right. So remember my model. This is what my model looks like, right? So what are my variables? I've got lots of the different concentrations of these different polymers floating around, right? So I could look at a time series of these concentrations, which is what these are, right? Darker colors means there was more at a given time. Lighter colors means there was fewer. Time goes that way, that direction. OK. And I could say, all right, this is one sequence, and this is another sequence. How correlated are they? What's the mutual information between these 2 sequences? I get a number, right? Great. Simple number, throw it in this equation, comes out the other end. I can do this for all possible pairs of sequences in my system, right? Up to length 6, because I don't want to do it forever. All right, and then I can build something like this, right? So this is all these numbers where I've calculated what would happen, what the mutual information between them would be, right? So now I've got a weighted network, right? This is an adjacency matrix for a weighted network where the weights on the edges are the mutual information shared between those two sequences. And the nodes are the sequences. This is capturing the dynamic correlations in this complicated model, right? So this is what this model looks like. This is what the correlations look like. Now, I've got the stochastic block model, so I can ask, is this structured? Is there structure there? Is there a rigorous way to characterize it? And the answer is yes, sometimes. So depending on how effective my catalysts are, structure emerges or it doesn't in this system, right? So this is, I used a different color scheme. I'm sorry, I made all these slides yesterday. So these are the same networks that I just showed you. This is when the catalyst increases the reaction rate by a factor of 10. And this is when the catalyst increases the reaction rate by a factor of 1.1. It says 0, but that's a typo. Right, so in this case, if I use a Bayesian inference method, I can be pretty confident, my belief can be almost unity, that there is structure here. And the algorithm kind of breaks it up into this core periphery structure here, which is nice. The same thing for the low catalysis rate. My belief is like barely better than 50-50 chance. I'm like, no, this is just, it's probably a mistake. Like, there might not be structure there at all. It's 50-50, who knows, right? So these are two limits. OK, so now what I can do is swing between these different catalysis rates and figure out what the likelihood of structure being seen there is, which is what you see here. Man, that came out really bad. OK, all right, so this is basically relative belief. How much stronger do I think, how much more should I believe that there is structure there than I shouldn't believe? So 50-50 means like, it's toss of a coin. You can't really be sure. Here is the catalysis rate. So I started at 0.1 here, and then all the way to 10. You notice like it stays pretty constant all the way till about 5, 5.5, and then you get this sharp upswing here. This is actually very well known in a very different context. So people have seen phase transitions like this in Ising models. They've seen this in the community detection literature in network science. So I can't be sure yet because I made this yesterday, but I'm pretty sure this is a proper phase transition. So what do I even mean by that? What does this phase transition represent? I'm running out of time, so I'll try to talk quick. What do I mean? All right, do you guys know what Bernard cells are? Like if you take a thin layer of fluid and you heat it on the bottom and like for a while nothing happens, but you turn it way up and then you start to make these like cycles right here, right? It's the onset of convection instead of just conduction. That's what's happening here. This is order on the scale of the dynamics in the system, right? This is disordered. This is ordered. This is the transition from disordered to ordered in the dynamics. So it's an ordered phase transition in the dynamics. And that's all I got. Any questions?
All right, we got time for like one question if it's awesome.
Anyone? Anyone? Nothing? So I confused everyone.
You got one?
Oh, man.
I just kind of noticed that this catalytic increasing factor, that's something in this very abstract model that you could actually relate to the real world.
It is.
So could you perhaps go through and say like, you know, this catalyst, this enzyme is really efficient, so that would be more likely to cause more order in the system?
Right, so that's what's interesting, right? So even if you have catalysts, if they're not very effective, if you're in this regime, you're not gonna see this global scale change. Mm-hmm. But they don't have to be that effective, right? So the average effect of the catalyst is to speed up the like uncatalyzed reaction by a factor of 5 right here, which compared to the biosphere,
yeah, that's not
nothing,
much for
right. And here, where it was completely ordered, it was only a factor of 10.
Wow.
So yeah, so
try a factor of 1,000.
Right. Yeah.
Or a million.
Yeah. Oh, I did. This asymptotes. It goes. Yeah. So yeah, it's a good question.
Yeah, that's awesome. All right, thank you very much.
Oh, I'm going to plug one more thing. If you think that scientific publishing can be done better, and you think astrobiology can help do it, you should talk to me before we leave, because I'm really passionate about that.
Are you starting your own journal?
I'm open to anything. I'm going to try to get everybody on preprints first, and then we can put it wherever we want.
Let's do this.
Let's do it.